Hi, and welcome to another episode of Tom Kennedy Science, and I'm your host, Dr. Tom Kennedy. Now, today what we're going to talk about is the control of the cell cycle. Now, think about that, control of the cell cycle. So we know that the cycle goes between interphase and the mitotic phase, and we can further subdivide those into the G1, which is the first gap, S phase is where you replicate your DNA, and then you have your second gap, and then, of course, you go into the mitotic phase, which is separated into both mitosis, which is division of the nuclear material, and cytokinesis. And, of course, we know that mitosis includes prophase, prometaphase, metaphase, anaphase, and telophase. And then some cells can actually exit the cell cycle. And when they do that, they go into a quiescent stage, or I wouldn't really call it a resting stage because they're actually quite metabolically active, but they may also be trying to repair their DNA. And most of our cells are actually in this G0 phase. They're not actually in the cell cycle. And if a cell is in the cell cycle, it spends most of its time in interphase. The point of this whole lecture today is that this cell cycle, the cycle between interphase and the mitotic phase, it's regulated. I mean, it's highly, highly regulated. Think about this. You have 17, maybe even 20 or more trillion cells. That's amazing, isn't it? And each of those cells is an independent living thing. Well, not completely independent, but each cell is a unit of life, right? And you're a conglomeration of all of these cells, and they have to be told, when do you grow? When do you divide? When do you commit suicide? When do you come out of the cell cycle and just maintain yourself like a nerve cell potentially for decades? So the point here is the cell cycle is under tight control because you have to have all of these cells regulated so that you can exist as a multicellular organism, so one or more cells just don't take over. And in fact, cancer, that's what happens when we have cancer, right? Cancer it's horrible. So it's a dreaded C word. We all dread it. We all know people that have gotten cancer. But cancer is really the price we pay for being a multicellular organism, because cancer cells are damaged and they have escaped the cell cycle. In my previous lecture, we talked about the cell cycle. Now, let's talk about how that cell cycle is regulated. So, depending on the type of cell, the frequency of cell division will vary. You have epithelial cells. These are the cells of the lining of your body, like your cheek cells or your skin cells. You're losing 50 billion of these a day. They don't last very long. They're rapidly going through the cell cycle. You have other cells, like a nerve cell, once you're an adult, may never grow and divide again unless it becomes damaged, and then will come back into the cell cycle and repair itself. Or you might have a liver cell. They might divide once a year. Okay, so the idea here is that the frequency of the cell division is dependent on the type of cell and the differences in the cell cycle. How fast are you going through it? Like I've got a wound, and those cells are rapidly repairing as fast as they can, I promise you. And also something like your liver cell might replicate once every other year or so. The speed at which these things are replicating are the result of regulation at the molecular level. So let's start taking a closer look here. We got these cell cycle, and we know that when you're in interphase, you've got your first gap, which occurs between the mitotic phase, M phase, and DNA synthesis, which is S phase. And that first gap has a checkpoint. And there's another checkpoint at G2. And there's another checkpoint. There's actually two checkpoints in the mitotic phase. One as you transition from metaphase to anaphase, and there's a second one in anaphase. So for the cell to complete the cell cycle, it's got to get through these checkpoints, okay? And this is when they can exit the cell cycle. So if you're in G1 and you either don't get a signal or you don't pass your checkpoint, you might go into a G0 phase where you either repair your DNA or, hey, that's where you exist for the majority of your life, depending on the type of cell. Now, we said that the cell cycle, that is controlled by chemical signaling. All right, so it all comes down to chemistry inside of our cell. I know, a lot of people are like, I'm in biology. Why am I learning all this chemistry? Hey, chemistry is, it's at the heart of everything. So we have this protein complex that occurs inside of cells. It's called the MPF, for mitosis promoting factor. MPF, huh? And MPF is actually in constant levels throughout your cell. However, its activity does not stay at constant levels, and its activity is regulated by two different things. This MPF is actually a conglomeration of a couple different molecules. So it's a complex, right? You've got a cyclin dependent kinase. Cyclin dependent kinase. That means this kinase, which is a molecule that phosphorylates things, is dependent on cyclins. So we have another class of molecules called cyclins. And cyclins get their name because they go up and down in concentration throughout the cell cycle. And of course, there are different types of cyclins. So that's just a broad name for a family of proteins. And then also the second thing that regulates the mitosis promoting factor are the presence of phosphate groups. So your CDK, your cyclin-dependent kinase, its activity depends on whether or not it's phosphorylated and whether or not it's bound to a cyclin. Okay, so let's take a look at this. Cyclin-dependent kinase. You know right off the bat, a kinase is a molecule, it's an enzyme that phosphorylates other molecules. So whenever we're talking about a mitosis promoting factor, to go into mitosis, for the cell to divide, you have to activate a lot of different things, right? There is a lot of other molecules that must get turned on. And the way we often turn on molecules is we phosphorylate them. Okay, so a kinase is going to go out and phosphorylate them. So this cyclin-dependent kinase, when it becomes active, it's going to start phosphorylating all these different molecules. Now, it's only active, or for it to be active, it has to be bound to a cyclin molecule. And like I said, there's different types of cyclins, but specifically the cyclin, when it's high in concentrations, it will bind to the CDK. Okay, now this is the whole thing is this MPF now. Now, interestingly, cyclin levels are going to rise prior to mitosis, but we don't want the cell to go into mitosis too early. So if we started having cyclin levels rise, all these MPFs would become active, the CDKs would start phosphorylating things, and we would kick the cell into mitosis. And we don't want to do that until we're ready. So there's a second way we regulate MPF, and that is with a phosphate group. Okay, so there's you got two different phosphate groups on this one, but you have what's called an activating phosphate group or an inhibitory phosphate group. So we got activating and inhibitory. So we do not want CDK to phosphorylate things until the cell is ready. And when the cell is ready, there'll be lots of cyclin, all bound to the CDK, so it's ready to go. But then there's that inhibitory phosphate. Yes, you can actually turn off molecules with a phosphate. It does. You can do that. It all involves shape change. Then right when you go from G2 into mitosis, or the mitotic phase, what's going to happen is that there's going to be a protein that's going to become activated. I know, another protein getting activated. And then it's going to remove the inhibitory phosphate. And guess what? MPF will now become active inside the cell. So at the end of G2, once MPF is totally active, it's going to, like I said, it's going to phosphorylate lots of different molecules. And by phosphorylating lots and lots of different molecules, it will initiate mitotic phase. And then what happens next is, well, this thing has to be turned off, right? And in fact, if your MPF doesn't get turned off, the cell's going to keep trying to divide all the time, right? So the cyclins during anaphase, they're targeted. They're targeted by something called a proteasome that breaks down the cyclins. Hence, their numbers begin to decline. And as the amount of cyclin in the cell begins to decline, the CDKs become less active. And then by the end of mitotic phase, they're starting to get the inhibitory phosphate back on them. So now all of a sudden, our MPF is now turned off. Okay, and then we're out, and then we're back to G1 again. Now, that is how a mitosis promoting factor works. That is one of the many chemicals involved with the cell cycle. Now, what I'm going to do now is talk about these various checkpoints. There's a G1 checkpoint, a G2 checkpoint, and a metaphase or mitotic checkpoint. And like I said, there's another one in there, but I'm not going to go too much into the anaphase checkpoint as well. But what I am going to talk about are these checkpoints, especially G1, G2, and the metaphase checkpoint, because these are really important. And what these checkpoints do is they prevent the cell from progressing through the cell cycle if there's a problem. So you've got to pass by these checkpoints. Okay, now cells will sometimes get slowed down. The checkpoint does its job. There's some DNA damage. Chromosomes didn't get separated correctly. The cell at this point will attempt to correct that damage by repairing the DNA. And if it can't, under normal circumstances, the cell will commit apoptosis, right? Because if it doesn't have the right DNA and it can't fix it, or it doesn't have the right chromosomes, it may not be able to survive anyways. And the cell will commit apoptosis, sorry. And that is a way that, cells can safeguard themselves against cancer. Okay, so let's dive deeper here. One of the most important checkpoints, they're all important, but this is a really big one, is a G1 checkpoint, or the first gap. Now, in animal cells is what we're talking about here mostly. In an animal cell, for you to proceed from G1 into S phase, that's your DNA synthesis. If you can pass this checkpoint, you've committed to the cell cycle. Because by committing to the cell cycle, by passing this checkpoint, you are going to now start replicating your DNA. And that's a big energy investment, right? Because you've got to create all the proteins necessary to replicate your DNA, spend the time replicating your DNA, you're immediately going to go to G2, where the cell will want to go into mitotic phase so it can duplicate itself. All right? So the G1 checkpoint, this is big. Like I said, it commits itself to the cell cycle. It's going to spend all these resources duplicating itself. So that's what animal cells are really all about. And for an animal cell to pass through the G1 checkpoint, the first thing it absolutely has to have, you can't do it without this, you have to have a chemical signal outside the cell. There's all kinds of terms for this. Scientists love to use fancy words like exogenous, which means just outside the cell. I just prefer the term growth factor because that's telling the cell to grow and divide. Actually, the cell might not be growing, but to divide. Or a mitogen, which is another one of these names for a chemical signal that causes the cell to go into mitosis. So you get the idea. You're getting a chemical signal. The cell has to receive this signal that says, you need to go into S phase. You need to complete your cell cycle. So that's the very first thing that a cell needs to get is this chemical signaling. And I think Freeman's biology calls this social signaling or social cues. And you're just receiving a chemical signal, right, from outside the cell. And a lot of these growth factors, they bind to RTKs. Remember RTK? Receptor tyrosine kinase. Man, another one of these kinases, right? And what they do is they receive these growth factors and they transmit the signal into the cell. And they do that by phosphorylating their tyrosines, which then activate all these other molecules, which initiate the cell to go into mitosis, right? So like I said, that's important. It's helping you get past this G1 checkpoint. In animal cells, you must get that chemical signal to proceed to S phase, all right? That's normal. And if you're proceeding to S phase without a chemical signal, you're damaged. That's a cancer. That's a cell that could be potentially cancerous. Other stops here for the G1 checkpoint. In animal cells, it's mostly about DNA. If your DNA is damaged, then the cell might come out of the cell cycle and attempt to repair that DNA. And so it's going into a G0 phase to repair the DNA. And if it can't repair it, the cell will commit apoptosis. And if it can repair it, it comes back into the cell cycle. Now, interestingly, have you ever heard of like tumor suppressors or tumor suppressor genes? Okay. A tumor suppressor gene is a gene that makes a protein involved with these checkpoints. And one of those proteins is called P53. Now, P53, this gene, if it becomes activated, it can stop the cell cycle until the DNA is repaired, or it can trigger apoptosis. So if you have damage to the P53 protein gene, right, then you create a damaged P53. That means the cell might not get kicked out of the cell cycle and will continue to replicate its DNA even though it's damaged. This is exactly what happens to cancerous cells, right? So you're starting to see I'm leading up to some cancer here. So yeah, tumor suppressor genes are very important. You're getting through this G1 checkpoint. You got your chemical signal. You have no damaged DNA. Now you're in S phase. you replicated your DNA, or you hit G2 now, the second checkpoint. This is where a lot of cell growth occurs, getting ready for mitosis. But next is a G2M checkpoint. And you can imagine if you just replicated your DNA, what are you going to check for? Damage to your DNA, right? So the G2M for the mitotic phase checkpoint, the cell is going to check and try to attempt to repair DNA damage. And once again, P53 is involved with this checkpoint. So if you get that gene damaged and your P53 protein doesn't work, then the cell will continue onto mitotic phase with damaged DNA. All right, so now you've gone through the G1 checkpoint, you've gone through the G2 checkpoint, you are in mitosis, or specifically mitotic phase, right? There is a very important checkpoint during mitosis, and this is the metaphase checkpoint. If you remember, we split mitosis into prophase, prometaphase, metaphase, anaphase, and telophase. Okay, now metaphase, remember, all the chromosomes are lined up at the metaphase plate. This is our checkpoint. Because if the cell cannot line up the chromosomes correctly, all right, if it can't arrange them to the middle of the cell, then when it transitions from metaphase to anaphase, if it can't line those chromosomes up, you could have a non-disjunction. You could have a problem where your chromosomes don't separate correctly. And that would be detrimental to both daughter cells, right? Because a daughter cell might be missing a chromosome, or it might have too many chromosomes. And both of those can cause problems for the cell. All right. So there is all of this tug of war back and forth between the kinetochore microtubules. Remember that? Those are the microtubules that attach to the kinetochores on your chromosomes. And there's this tug of war back and forth to get those things lined up. And there is an enormous amount of chemical signaling going back and forth between the kinetochores and other parts of the cell to make sure that these chromosomes are lined up correctly. I mean, it's astounding. And then, if it gets it lined up, then guess what? There's other chemical signals involved. And what these do is, and then if they can get them lined up and passed by this metaphase checkpoint, then the cell will release other proteins that will begin to degrade things called cohesions. Cohesion, kind of holding things together. Cohesions hold the sister chromatids together. Imagine little scissors going in there and cutting up the cohesions and allowing the sister chromatids to separate, and the cell will then go into anaphase. And importantly, a lot of cancer drugs, they specifically target the cell at metaphase and attempt to prevent it from passing from metaphase to anaphase, which is kind of cool in itself. Now, one thing I want to share with you that I think is really cool is this is anaphase, and these are all the different genes involved with anaphase. I know we're, your first year biology class, you look at some slide with these chromosomes. Oh, yep, those chromosomes are in metaphase, those chromosomes are in anaphase. And we sometimes forget the complexity of a cell and the choreography of all of these genes working together to create proteins for a certain specific task. And I just love looking at this chart right here with all of these different proteins involved with anaphase. And of course it says genes involved because genes code for proteins. My family was coming in to join me here for a little lesson on mitosis here. It's fantastic. My daughter is like 3, right? And she wanted to come in and see what daddy was talking about. All right. So getting back to this is just kind of showing the complexity of just one phase of mitosis. And to me, that's really cool. So there you have it. That is a very brief introduction to the control of the cell cycle, right? And we talked about one particular type of mitosis promoting factor and how the cyclins interact with that. And then we talked about the cell checkpoints, how you proceed from checkpoint 1 into S phase, G2 checkpoint, and then the mitotic phase, and then the metaphase checkpoint as well. And the idea here is that the cell must pass each one of these checkpoints to proceed through the cell cycle. And there's ways that the cells can come out of these cell cycle, especially if there's damage to the DNA. And the problem is when there's damage to the DNA and the cell cannot fix it, under normal circumstances, cells will commit apoptosis. if they can't fix it. They commit suicide. And what we start to see with cancer cells is a couple different things. We see an evasion of the cell cycle. They can bypass these checkpoints. They replicate with damaged DNA, and they don't repair it, and they don't commit apoptosis. So we can think of cancer in several different ways. One is a failure of apoptosis, a failure to commit suicide, and a failure of the checkpoints to stop these cancer cells from growing. Okay, so there you have it. There in a nutshell is regulation of the cell cycle. Of course, it's way more complicated than what I talked about here. This is just kind of like in broad strokes how this works. Until next time, it's been another episode of Tom Kennedy Science.